Got it. Yeah. Mhm. Yeah, thank you for the intro, Sophie. Yeah, happy to see some faces here in these vacation times. So structural proteomics is probably one of the smallest unit in the SciLifeLab community. Um, but what we're doing is that we're providing cutting-edge equipment expertise for protein-protein interaction analysis, and that combines pretty well with all the other stuff that you see, some stuff going into a funnel here, which we call like integrative structural biology. So I'm part of a platform in SciLifeLab that's integrated structural biology. And there we have NMR, cryo-EM, and all the other stuff virtually associated with this. High resolution techniques, low resolution techniques, and then structural proteomics, which I would call middle resolution technique. Basically, with HDX, we can get some dynamics, and with cross-linking, we get some orientations and stuff like that. So at the moment, this is some background infrastructure things. So as I said, we're combined with NMR, which have centers in Gothenburg and Umeå, and is coordinated from the NMR center in Gothenburg. And then we have also the cryo-EM, which we're virtually attached to, which is in, of course, Umeå and Stockholm, and now also in Lund with the microscope out at Max 4. And we have regular meetings, super nice people, and there is basically a lot to gain for collaborations in these settings, actually. So there is a lot of projects flowing from different units here, and a lot of talking and meetings, which are super fun from a scientific perspective for me. I've been to Umeå and done cryo-EM myself, never done it before. It was super fun. We started out, though, before this, we started out as a bio-MS entity, basically like a small HDX mass spectrometry, just capacity in bio-MS. And that started in like 2017, 18, and I was despairing a bit. So, because we had basically no projects, we were only meeting with mass spectrometry people, and they were not like super interested in this somewhat niche technique. They didn't understand it, just thought it sounded very difficult, and well, it isn't. But then Johan and I made an application to SciLifeLab when they renewed 2000, year 2000, and then was also this COVID crisis at the same time. So up to that point, basically 2000, 2021, we had like 7 PIs that we serviced every year. So maybe 7, 10 projects per year. Not, I mean, we had something to do, but it was not so fun for us that worked there. It's me, Lotta, and some other people. But then when we came in there, suddenly we went to the right meetings and met up with structural biologists, and now projects started to flow in. So since then, now that this graph only stretches to 23, but we're maxed out basically at this point. We do service like 30 PIs per year, maybe 20 HDX and 10 cross-linking projects per year. Some of them small, like one protein against another. Some of them huge, like 16 proteins or something like that, crazy. So basically now we're running flat out. And I also think that we have a lot of luck with both projects. We have success rate of 75% basically for our projects. Some nice publications with high impact factor and all that nice stuff. So basically what we supply, it's like a full service. So you come, you leave the samples, and it's a very close collaboration with the project, and we basically deliver out finished data. It's not just an Excel sheet. There is analysis, discussions, figures made for publications and so forth. But we do sequencing, mass spectrometry, of course, like de novo sequencing of antibodies. You will hear more about all of these techniques later. I will not talk at all about this. And we can do affinity mass spec and limited proteolysis on demand, not something we're very often asked to do, because what people ask for is the HDX mass spectrometry and the cross-linking, and that is our main bread and butter, so to speak. There is some other forms of mass spectrometry that we could do, but either on demand or if we get more people in. I'm recruiting at the moment. Yes, so we have, of course, one can go to our homepage at SciLifeLab, and there you can see all the nice publications that people who have been using our services actually publish. It's not our idea, but you get an impression of what we work with. Some very, very talented people. And these are just examples of projects from Lund. It's targeting toll-like receptors with autosmitchen. We have some work with Derek Logan, Vasily, and also Christian Bellodi, which was very, very, very nice work. With that, one of the best things for me being an infrastructure is actually not only being able to help researchers getting nice results and understanding their hypothesis, but it's also collaborating internally. And when I started, it was a little bit empty. We didn't have Lucas, for instance. And Johan and I, of course, has a group, mass spectrometry, next doors, and is our boss, our main boss. And we had, yeah, we should do this and that, but no time to do something. But suddenly, one of his PhD students, he started to show up in my office more and more. And yeah, could you teach me? Could I use this machine? So super nice for me. And I basically, we did some test runs together, and you were walking next to me, running some stuff, and then you were able to do a lot of stuff yourself and using the machines while I was on vacation or we had some dead time. And actually, they made a lot of nice work. I think you actually had HDX, and at least in all your papers in your thesis. It's crazy. And it's a very, very nice thesis that was awarded best PhD thesis of the year here at medical faculty. So it's with great pleasure I would like to hand over to Dee, who will talk a little bit about the work he did when I didn't use the machines.
Thank you. All right, let's move to my slides. Yes. All right. Thanks for a very nice introduction, Simon. So today I'm going to present my talk. So the cover is actually the three most important molecule in my PhD thesis. So try to render it nicely with this material-wise like. So those are the three discovery I finished study and published with Simon, of course, with all the collaborator during the past PhD study. So my title is Rethinking Integrated Structural Biology, What Structural Proteomics Bring to the Table. So I will quickly go to the integrated structural biology concept and what structural mass spec encompassed. And I will work with actually three of my projects to walk you through the journey where I feel like this is the time we can apply structural MS to try to provide some very unique molecular insight. And we'll do a quick summary and some take-home message. That will be today's presentation. So the integrated structural biologists try to encompass different modality of the structural biology methods. You should have pretty conventional biophysics method like X-ray crystallography, cryo-EM, NMR, but you try to have more complete and deeper understanding of the molecular events. But sometimes not all the scientific question fits into this category. They can give you something like X-ray crystallography, cryo-EM, MMR, you're guaranteed a structure, but sometimes the scientific question does not align well with that kind of techniques. So this is the time structural MS, structural proteomics, can be super helpful. So here's some pretty good example throughout the years people have been using to generate those structural proteomics data. So, but today's talk I will focus on HDX, cross-linking, a little bit of affinity purification mass spec. So of course, we're a mass spec lab, so I have a protein of interest. My protein of interest is the streptolysin node. It's a very important bacterial protein for the group A Streptococcus. So here you can see can annotating from structure at the 4 domain, but things to remember, some of the domain are discontinuous. So that's a little bit more requirement for the protein design. Actually, this protein is a secreted toxin. It works by first secreting monomer form in solution and bind to the eukaryotic cell line membrane and start the oligomerization, form the 40 to 60 mer ring-like pore and punch the cellular membrane, cause the cytoplasma leakage and ultimately cell death. So the primary function, as I said before, is the cytolysis, but they are also reported multifunctional role. But the interface, the molecular feature of those interactions has not been solved. So that came in my first project. We tried to map the streptolysin node human plasma protein interactome and try to find if we can get something really valuable in terms of the biological relevance. So we start with affinity purification mass spec. I'll quickly go through. So basically what you did is immobilize your bait protein and incubate this bait protein with the pre-mixture. This can be human plasma, this can be saliva, and you wash quite rigorously to get rid of all the unspecific binding or weaker binder, and you elute them. You use mass spec to try to identify and quantify the enriched protein. So in that case, you have basically a bait-prey quantification matrix, and you can do some pretty cool network analysis. This will give you something like this. You have the SLO as your bait protein. There are tons of interaction. They might be direct interaction. They also can be intermediate interaction, which means that there's maybe 3-4 protein assemble. So from this, you can actually rank. We actually find one protein, human plasminogen, which has non-reported before as this streptolysin node interactive partner from this affinity purification mass spec. But just prove the binding is not enough. We try to understand what does this binding actually mean. And I come up with this functional assay where we can clearly show that the streptolysin node, it does not directly catalyze this plasminogen activation to plasming, but in a dose-dependent manner, this SLO binding can accelerate the activation. Normally, our projects stop there because we don't have the resolution power to go further. And then I go to the literature. There are some people who do this conformational change of the plasminogen molecule, but they only manage if the binder is peptide binder or chemical region. But here we're working at this streptolysin node, the 4-domain 60 kilodalton protein is really big one. So in their original paper, this says this cannot be done with that bacterial binder. So what I do is I go to Simon to say, okay, if this actually fits, okay, we can always try. So what we do is we do this the hydrogen deuterium exchange mass spectrometry. Quickly go through the workflow is you have your target protein. You try to study its solution state behavior between different state. You have the apple state, which is without the binder. You have the bond state. Normally, you can have antibody, but in this case, the streptolysin node bacterial protein. And you deuterate them in the different interval. So actually, it can interfere from also the temporal scale. And then you quench the deuteration. You do the online digestion to the peptide, and you measure the peptide. You measure those mass shift. You have the baseline, which is on deuterated 1, and you actually take a look at this change of the deuteration to infer the actual solution state behavior. So in this case, we actually observe this very delicate but global stabilization profile throughout the interval. And from the most extensive interval, we can actually pinpoint certain protection region. In this case, it could be allosteric deprotection, and it could also be the direct binding involved interface here. So what are we going to distinguish that? We're going to apply another which, like Simon also said, nowadays reviewer just ask for more modality of the data to prove your point. So here we did a little bit preliminary test. We try to use crosslink to see if we can distinguish those interfaces from this allosteric effect. Can we pinpoint the binding interface here? So the cross-linking mass spec works like this. So you introduce this chemically reactive region that would only cross-link target residue if they are sitting in the close proximities in space. And this covalent bonds can stay throughout the process of the protein digestion, cleanup, and you shoot in the mass spec. And you can analyze both in terms of the intraprotein, which is the distant constraint within one protein, or the most valuable one in this case would be the interface, inter-crosslink distant constraint. So collaborate with our computational modeling expert, we finally generated this model, which is the best model, combine both the energy score and the distant constraint violation. The more magically, it supported independently down HDX data in terms of the interface. To conclude of the first paper is that we have a structural mass spectrometry-centric approach. We identify this streptolysin with a previously unrecognized role to bind in the plasminogen. In terms of the molecular insight, how to explain this enhanced accelerated activation, we managed to also illustrate that molecular insight using structural mass spectrometry and computational modeling. So here's an example of the power of the integrated structural biology approach. So when we map this, to overlay this complex with the streptokinase, which is known, has a co-crystal structure with the plasminogen, we find out that our interface does not overlap with each other. This also explains why this streptokinase also work in terms of the accelerated activation in the previous functional assay. So here's just the in vivo mechanism we try to propose from this study. So next, we move to our favorite part, is the antibody-antigen interaction. So we start this project because we know that one important biological feature of the bacteria is they have this immunohierarchy effects, which means a certain part of the antigen is more easily to trigger host immune responses, but in most cases, they can be non-functional. You want to target that subdominant part, but then you need the monoclonal antibody to help you pinpoint that. A very brief recall of the antibody biology is that you have this heterodimeric molecule, Y shape with two Fab arms. The tip of the Fab was formed by this six CDR region, both from heavy chain and light chain. The diversity of the CDR chains determines the antigen binding specificity. And you have the FC region that was further related to the downstream factor mechanism. But in our case, we most focus on this Fab region, how it interact with the antigen. But in the field now, to do the epitope mapping, the very straightforward way, or the most studied way, is to do this linear epitope mapping. This can be easily done, not easily done. You still need to generate a big library and do the screening, quite labor intensive. But in our case, or in most actually the case people find out in bacteria toxin particularly, those epitopes are normally conformational, which means that those interface residue, they are discontinuous in sequence, but they are close in space. So it has to fold in that particular patch to be recognized by the Fab. In that case, if you generate those linear peptide, it won't work. You do this mutagenesis, scanning mutagenesis one by one, it also won't work. So you have to capture that native antibody-antigen complex in solution state. So normally people do it with the co-crystal or cryo-EM, but there's a reason why the AlphaFold didn't learn too much feature about the antibody-antigen interaction, because there's so few co-crystal structure has been solved. So that's more challenged in a sense. And then it comes to this paper where we apply initially 3 different modality of the mass spectrometry approach to identify a protective epitope in the streptolysin molecule. This is also the first epitope has been identified for this very important gas, the antigen or vaccine targets. And to define the antibody production in this study, we only focus on this neutralization assay. So here you can say that if without any antibody or any functional antibody, the S-low protein would form a pore on the cellular membrane, and you have the cell lysis. But if the antibody is neutralizing, this process can be stopped. So that's how we define this monoclonal antibody from the first place, as the protective antibody. But then the problem is this antibody is actually a hybridoma generated clone. So we have no idea what the sequence is. So Simon helped me, and also we have Carlos, who was in-house the expert in de novo sequencing. We started to work directly with antibody digest with different enzyme and do this de novo sequencing and assemble to the full length of the antibody. Because now we're only going to model a focus on the Fab region. So this is only the Fab model of the structure, but you can see overall you have a pretty good confidence score. This is actually the log2 transform of the precursor ions that support the residue. So like each residue in the full length in the end has at least 8 precursor ion evidence. So that's pretty good quality in general. And then we did the cross-link again. That gave you this interlink. So let's have an animation here. Yes. So with that information, you can actually generate this nicely interaction space, give you a preliminary understanding of where this Fab is primarily located at. So you can see that the middle part of this toxin is actually targeted by the antibody. And then we do a more rigorous data-driven docking where we input both paratope information, epitope information, and cross-linking to drive to have this most representative model from the largest cluster of the modeling. This actually showed that in that particular domain 3 region, you have the epitope located. Again, we want more data to prove that if that's the legit. So now we redo HDX. In HDX, you don't need to know the sequence of the antibody. In that case, we also have a pretty good resolution and confidence to locate the epitope in that domain 3. So you can see here is the alpha helix with beta sheet and term beta sheet. So that's the epitope patch we finally identified that was supported by both modality of the mass spectrometry approach. But then we actually tried to prove this is actually the epitope. We managed, the colleague, they do de novo design to extract that modified domain, make this modified domain 3. And we use the same antibody, as we all know, highly specific, won't recognize the unspecific binding. It binds to the same pattern compared to the native, which is the full-length strep here. So to summarize in this paper, we figured out where the epitope is, and when we actually align with the previous published result, we actually see that it overlapped with this critical motif that was involved in this from monomer to oligomer transition of this streptolysin toxin. And more importantly, if you talk about the vaccine target, you want to make something that was highly conserved across the string. So my colleague, they query this epitope into the all known gas genome to find out that this is close to 100% conserved across all the genomes. That make a really good target if you want to generate the vaccine that can provide broad spectrum protection. So the final part is, now you want to make a vaccine, but if the protein is, your antigen is very small, normally it won't work. Immunogenicity, it's a biophysical property, it just won't work in general. So we turn to this cysteine viral-like particle modality that was initially developed in David Baker's lab. When they have this epitope fusion trimer and mix with assembling pentamer, so that would give them this icosahedral self-assembling 2-component protein-based nanoparticle. So this has been shown in several studies, has several beneficial in terms of the size is the optimal size, that it stays long enough in the lymphonoid, but not too small to flush out too quickly. So that increases the chance of this nanoparticle to meet with immune cells. And also this multivalency has been shown to enhance the crossing of the B cell receptor that further increase the activation signal. So we now have our antigen. We want to use this antigen presenting delivery system. That's what we did in this paper. So it's now in bioRxiv with some preliminary data. I will show you some highlight of the results. I further try to understand the vaccine study using structural mass spectrometry. So again, because we didn't solve the structure of this modified monomer, we want to see how close it is compared to the native domain 3 from the full-length streptolysin. Also, here I think HDX is a super relevant approach to capture the solvent accessibility, also to show that there's no misfolding. You might be something a little bit different in terms of in the C termini, the have a helix region, but overall it's quite the same. The order of the secondary motif also follow the same. And now we know it works, so we start to make this fusion component. We mix them with assembling component. We have this icosahedral, but this is just the predictive model. Then we actually do a second mouse to show that, okay, you do have a highly stable and monodispersed antigen-loaded nanoparticle vaccine. And we take this fraction particularly, and we put in the cryo-EM that gives you this very nice separation of the particle. And then this is some ongoing work. We try to do the cryo-EM reconstruction. You can see the scaffold region has been nicely resolved. You can appreciate those secondary structure, but the antigen part that was presented on the surface is a little bit fuzzy. That needs more work with this because there are some asymmetric mismatch. And in general, those antigen, because naturally they're not trimer, so and we put some flexible linker that give a lot of this heterogeneity here. But still, at this stage, you can still compare monomer in different format. So in monomer form, in trimer, it's a little bit destabilized. And if you assemble into another particle form, you start to be more stable again. And that the last one in another particle group, we actually shoot the massive 2.8 megadalton particle in the HDX. That tells you something like dynamic range the HDX can handle. Of course, because it's a repeated region, normally that will work. That does not increase the sequence complexity, essentially. And then we have some preliminary data to say, okay, if this whole system work out, we have this immunization cohort when we immunize mouse with different construct, and we sample it in the final time point, give us the very high titers. That give us a lot of confidence. I think this is particularly important in terms of the vaccine study, that not only look at the result, but you have to be assured your sample quality is legit. I think this is the problem currently in the Streptococcus vaccine field. People just produce tons of recombinant full-length protein and mix them together, call it multi-component, but they never check the biophysic characteristic of their formulation. Some of the protein tends to aggregate immediately in even no concentration at all. So you have to understand the vaccine sample formulation quality. For me, it's very important. So the summary is we review this multifunctional role of the streptolysin, including the previously unrecognized function to enhance the plasminogen activation by this structural MS-centric approach. And we demonstrate that multimodal protein mass spectrometry is very useful in epitope mapping, especially for conformational epitope. And the last, we show this first antibody-guided epitope-centric nanoparticle use case for the bacterial infection, and also with the sample quality assured by the HDX-MS. So the take-home message, pretty quick, is the mass spec, like Simon said, we're a pretty niche field in terms of structural proteomics, but I think it's the fast advancing, fast basing. We can do tons of stuff before we shoot in mass spec to gain the normal ideal quantification result. And the information we can harvest here can be super useful to complement those integrated structural biology concept. And several things that I try to reflect is the overall turnover time, I think, is pretty fast in terms of the sample prep, actually running the experiment. To interpret the data might take a little bit long time, but normally it's quite fast. and does not require milligram level, super pure 99% of the protein you want. So that can be a really good for initial screening or like initial stage of your research project. And structural MS, as I showed before, doesn't give you the structure, but it shows you some very unique complementary molecular insight. This case, in our case, is more like solution state, this population level, like cross-link. You can, if you cross-link a mixture of protein, you kind of capture all the conformation of the protein in that sample. So those are very helpful for you to understand the molecular state, the solution state of the molecule. And it's ideal for some certain scientific question that was probably suited for other conventional biological tools like NMR. I believe they cannot handle protein too big. And cryo-EM, they ask you to have at least 20K Dalton. That's your limit. Below that, if there's no very symmetry feature, they'll be super hard. Also, the flexibility really matters. Crystal structure, I've been there. It's very hard to crystallize even a fab alone. So let alone to saying the co-crystal structure of the full-length antigen with the fab. I mean, I think that's the problem. I feel like structural MS can be super useful in that sense. Of course, the last but not the least is this integrated structural biology. Seems to become even more popular right now, especially go with AlphaFold, all those Rosetta docking tools. But they really matter. They really need this informative distant constraint. So this can be generated very nicely by cross-linking or even more tight distant constraint by HDX. So in that, I want to summarize the take-home message. You can forget about everything I just said, but if you have a very nice project, you can think about capitalize that, talk with Simon and Lotta, and you won't regret. Last but not least, I want to thank the group, of course, the PI, Johan Malmström. Special thanks for Lotta Happonen and Simon Ekström, really teach me how to do this beautiful structural mass spectrometry, and everything just come out of it. That will be the end of my presentation. I'll be happy to take any question. Thank you.